Welcome to the Biology 260 lecture in microbiology. You are able to access these notes to print them out so that you can go ahead and just take additional notes on these papers if you would like, or mark any other notices on here as you go through. You'll notice that it says on here corresponds to Bauman Chapter 1 or Tutora Chapter 1. Both of the textbooks are actually very similar. There is just a couple places where the chapters are different, so I went ahead and noted it for both on here. So if for some reason you're using a different book than what's assigned for your school, not a big deal. You can see which chapter it corresponds to here with the lecture. This first lecture is going to look at an overview and the history of microbiology, looking at some of the historical figures. There are little video clips in here that I recommend you watch. I can't actually play them during my lecture that I am recording. When I put them on YouTube, if I embed another video on here, it will just delete my video for copyright purposes. So in your notes, I do recommend that you watch them. If for some reason the videos are no longer working, let me know. I don't actually own these videos, so if the person who put them up chose to take them down, there's not a lot I can do about it. I try and find other alternatives, and at the time I make this, everything is current, but that could change at any point. So if you let me know, I do the best I can to work around that and give you other alternatives, but most people find them helpful to look at. Robert Hooke, who was the first one to report life's smallest units were cells, did this in 1666. This was the beginning of the cell theory. The cell theory states that all living things are composed of cells and all cells come from other cells. The next significant person in starting the field of microbiology was Anton von Leeuwenhoek. He lived from 1623 to 1723. So in that time frame, that's actually a really long lifespan. He was a Dutch lens maker and discovered the microbial world. He called them beasties. So a protozoa he saw in 1674 and bacteria in 1676. Carolus Linnaeus, he was around from 1707 to 1778. He developed the taxonomic system that we call it the Linnaean system. Today we still use a modified Linnaean system. This is used to name plants and animals and group them together. So microbes or microorganisms are all organisms that are too small to see without using a microscope. So they have to be viewed with a microscope. Fungi are eukaryotic cells. They have a membrane-bound nucleus, and they're going to have cell walls. Fungi get their food from other organisms. They're going to absorb the dead organisms. We categorize a lot of the organisms based on how they eat. Within the fungi, we have molds, which are multicellular. They grow as a filament called hyphae. When you picture mold on bread or mold on cheese, it looks kind of fuzzy in appearance, and that's because of the hyphae. So yeasts are unicellular. They're oval or round, and they reproduce by budding. Protozoa are single-celled eukaryotes. They typically live in water, and some of them will live in animal hosts. Algae can be unicellular or multicellular. This group of microbes is photosynthetic. Prokaryotes are unicellular. They lack nuclei, and there's two groups that are categorized by their means of locomotion. The bacteria, they may have cell walls with peptidoglycan. The archaea, they tend to live in extreme environments, and they don't have peptidoglycan. Microbiology also includes the parasitic worms. The parasitic worms themselves are not microscopic, but they're often diagnosed through microscopic findings looking at their eggs. The idea of spontaneous generation, or abiogenesis, was the idea that life arose from non-living things. And there were several people that were involved in helping to disprove this idea. Francesco Redi, he was around from 1626 to 1699, was an Italian. He showed that decaying meat isolated from flies did not get maggots. This was helpful because the maggots had to come from other living things, the flies. John Needham was around from 1713 to 1781. He was British. He used these boiled and sealed gravy infusions and observed that they were cloudy days later. His work actually it led him to believe that this was attributed to a spontaneous life force that caused these gravy infusions to become cloudy. Lazzaro Spallanzani was another Italian. He was around from 1729 to 1799. He used these boiled infusions that he boiled for almost an hour, and the closed vials stayed clear. Those exposed to air became cloudy. So this was showing that there was an outside source that actually brought life into those vials. Louis Pasteur, he was around from 1822 to 1895. His work was similar to Spallanzani. He used a curved neck glass so that it still had air and all of the nutrients, but there was no spontaneous generation. Pasteur's work actually contributed pretty significantly to things we use today. He discovered anaerobes ferment grape juice, which is how wine is made. The process of pasteurizing things, where you would heat grape juice enough to kill the bacteria. A lot of things are still pasteurized today. You heat it up very quickly enough to kill the harmful bacteria, and then quickly cool it back down in the food industry. This began industrial microbiology or biotechnology, using microbes to manufacture products. Rudolf Virchow in 1858, he challenged the case of spontaneous generation with the concept of biogenesis, the idea that living cells only arise from pre-existing cells. The scientific method, this should be reviewed for you, is a technique for observing and answering scientific questions. Theories or law are going to be accepted hypotheses. They've been verified by many scientists in many experiments over the years. So these are well-studied ideas that scientists can rely on. When doing studies, we will often have a control group and an experimental group. The control group is going to be the one with a known outcome. No manipulation is done to them. The experimental group is going to be the one receiving the experimental treatment and will have an unknown outcome. It's important to have that control group so that you can do a comparison of what would have happened if you had not manipulated the situation. Edward Buchner was a German scientist who was around from 1860 to 1970. He showed that fermentation does not need living cells. He did this by demonstrating the presence of enzymes, which began the field of biochemistry. The role of biochemistry, it started the study of metabolism. Metabolism is the sum of all the chemical reactions in an organism. When we look at what goes on in cells and in any biological process, if you trace it back far enough, biochemistry is behind the explanation and the reason everything happens. In 1857, we have the germ theory of disease. This is the hypothesis that microorganisms are responsible for disease. The pathogen is going to be the specific organism that causes disease. Etiology is the study of the causation of disease. Robert Koch is one who is well known for the germ theory. He was around from 1843 to 1910, another German scientist. He investigated etiology. He studied infected animal blood and showed that given pathogens cause specific diseases. As a result of his work, he came up with these postulates that are known as Koch's postulates, that are steps to prove the cause of any infectious disease. First, the suspected agent must be present in every case of the disease and not in healthy hosts. The second is it must be isolated and grown outside the host. The third, when given to a healthy host, they develop the disease. And four, when the same agent is isolated from the disease host, it can be then put into the healthy host and it will be present in that experimental host as well. There are some steps to Koch's postulates that are not necessarily practical or ethical to use. So it is impossible to verify that the suspected agent is present in every case of disease and not in healthy hosts. It's impossible to test all of the healthy hosts to rule out that it absolutely is always not present there. Some things are not grown well in culture. So some are grown very easily on a petri dish, others have to be grown in chick eggs, others have to be grown in vitro. They have to be grown in a living host. They're just very delicate and don't do well in the lab. When this third one here is definitely a big ethical one that we don't use in identifying pathogens. It is unethical to give a healthy host a disease. To knowingly cause harm to a healthy person is not ethical in scientific research. So this is a lot of times studied in animals, but it's not able to be studied in humans. So then when the same agent is isolated from the diseased host and present in the experimental host, if we can't actually give them to an experimental host, we can't verify step 4. But they do give us a foundation of how we go about looking at is something infectious in nature or not. There were several other scientists that contribute to the golden age of microbiology. Charles Laveran was around from 1845 to 1922. He showed the protozoan cause of malaria. Edwin Klebs, he was around from 1834 to 1913. He described the bacteria that were causing diphtheria. Dmitry Ivanovsky and Martha Bejerink, around from 1864 to 1920 and 1851 to 1951, showed the disease in the tobacco plant passed through a filter and was a virus. So that's significant with viruses. They're very difficult to stop. They generally do pass through filters. There are a few filters out there that can catch viruses in them, but not many. Walter Reed, he was around from 1851 to 1902. He proved in 1900 that viruses cause disease. Christian Graham, he was around from 1853 to 1938. He was Danish. He developed a very commonly used series of stains or dyes that separate bacteria into two groups, called the Graham stain. Ignaz Semmelweis, he was around from 1858 to 1865. He was from Hungary. He required hand washing with chlorinated lime water to eliminate cadaver particles carrying disease. His contribution was significant because this was starting to look at these particles as being something outside the body causing disease, and first looking at hand washing. Joseph Lister, he was around from 1827 to 1912. He was English. He was the founder of antiseptic surgery. He used phenol or carbolic acid. So while these ideas of antiseptic surgery we kind of take for granted these days, that why wouldn't you wash the equipment, back in the early days of medicine, the idea was more on the lines of why wash this stuff? It's just going to get dirty when we use it on the next patient. Florence Nightingale, she was an English nurse around from 1820 to 1910. She introduced cleanliness and aseptic technique into nursing. John Snow, he was another English scientist around from 1813 to 1858. He studied cholera and contaminated water. He helped to document the need for clean water and sewage treatment, which ended up being a foundation for infection control and epidemiology. So having clean water and how we handled our sewage made a huge difference in how many infectious diseases were in the public health. Epidemiology is the study of occurrence, distribution, and the spread of disease. Edward Jenner, he was another English scientist around from 1749 to 1823. He was involved with the study of immunology and vaccination. He was around at the time of cowpox, and he used the cowpox pus to inoculate his son. His son got cowpox, but then became immune to it, which was a really important foundation for the idea of immunology and stimulating the immune system to be able to react to things in the future. Paul Ehrlich, around from 1854 to 1915, he studied chemicals that killed microbes differentially, but not people. This is an early foundation for chemotherapy. So hospital-acquired infections, they are sometimes abbreviated as HAI. You may still see them called nosocomial infections, since that was what people were taught for several years. These are infections acquired in healthcare settings. This is actually a very significant cause of mortality in this country. So depending on what statistics you're looking at, medical mistakes are sometimes up there at number 2 cause of death in the United States. Not all of them are infections, but infections are a significant number of those. Immunology is the study of specific body defenses against the pathogens. How do we protect ourselves? When we look at modern chemotherapy, we're searching for substances to destroy pathogens. Chemotherapy is the treatment of disease by chemical substances. Antibiotics are chemicals produced naturally by bacteria and fungi that are going to act against these microbes. So the golden age of microbiology is considered from 1857 to 1907, the work from Pasteur to Fleming with pharmaceutical microbiology. Sometimes they will almost look at it and say we're coming into another golden age of microbiology. Not that we're necessarily making so many new discoveries, but that we are going to have the field of microbiology change again significantly with microbial resistance. Albert Kluver and C.B. Van Niel, they were around 1888 to 1956 and 1897 to 1985. These people proposed that bacterial biochemistry was shared by all living things. This is actually very handy for us since it is a lot easier to study bacteria than it is living humans, and the biochemistry is indeed very similar. In the work of genetics, we had Oswald Avery and Colin MacLeod and Macklin McCarty. They determined that the genes contained molecules of DNA. So Watson and Crick, who established the structure of DNA, they did not know what DNA was, what its function was. George Beadle and Edward Tatum, they established that genes are actively related to the function of protein encoded by that gene. Molecular biology, this is going to combine biochemistry, cellular biology, and genetics. Linus Pauling in 1865 proposed that a gene sequence could provide understanding of evolutionary relationships. This is used more and more today to verify evolutionary relationships. Carl Weiss in the 1970s, he was a significant influence in determining that nucleic acids reveal three major groups: the bacteria, the archaea, and the eukaryotes. So some new areas of study in microbiology: recombinant DNA technology, or genetic engineering. This is looking at gene manipulation. Gene therapy, this is the process of inserting a missing gene or repairing a gene in human cells. Environmental microbiology looks to study the roles of microbes in the environment. So they are very handy for us in the environment. They're actually able to clean up a lot of mistakes that we make. Bioremediation is the use of living organisms to detoxify the polluted environments, which a lot of times are the result of mistakes that we've made as humans. So microbes are very important for humans. The normoflora are the microbes we live with our entire life. They live on and inside our bodies. They don't harm us, and a lot of them are actually beneficial or even essential for us to live. They help give us resistance to disease. The human microbiome is actually an emerging field of study. We're finding these microbes play a lot of other roles in our health than we previously thought. So we knew they were involved with digestion, but we are finding that they seem to play roles in our metabolism, potentially influencing cancer, some gene regulation, mental health issues, far more than we ever gave them credit for. Biofilms are aggregations of microbes. They can function as a group. Some of them can be harmful, such as bacterial endocarditis, or they can be beneficial to help buffer disease. So you have biofilms in your mouth, on your teeth, biofilms on your skin that help to discourage pathogens from becoming established in your body. Infectious diseases, these are just pathogens that invade a susceptible host. With emerging infectious diseases, these are diseases that are new or changing. They're either increasing or they have the potential to increase in the near future. So there are several of these. A lot of them may be regionally isolated. So if you go to a different part of the world, you may run into different emerging diseases. MERS, Middle East Respiratory Syndrome. There was 339 cases of this between April 2012 and June of 2014. It was found to be a coronavirus. SARS, the severe acute respiratory syndrome, is known for its outbreak in China in 2002. This is another coronavirus. Avian influenza, it's an H5N1 variation of influenza, sometimes called bird flu. In 2003, it killed millions of poultry and 24 people. In 2013, there was a different avian flu, an H7N9 variation that killed 131 people in China. Things that have been coming around gradually over the years. In the 1980s, we started to see the first cases of MRSA, the methicillin-resistant Staph aureus. In the 1990s, we started to see VISA, vancomycin intermediate Staph aureus, and in 2002 was the first VRSA, vancomycin-resistant Staph aureus. That's significant in there because vancomycin for quite a while was considered the drug of last resort. It was the only thing we had that we knew killed certain pathogens, and now they are resistant to that. So this resistance problem is continuing. Right now, we do have E. coli in the United States that are resistant to every single antibiotic we have. MDR TB, multi-drug resistant TB. This is resistant to at least isoniazid and rifampicin. West Nile encephalitis, West Nile virus, another emerging disease. BSE in 1996 is bovine spongiform encephalitis. It's mad cow disease. CJD is Creutzfeldt-Jakob disease. That's the human prion disease. The variant CJD is related to BSE. The variant is the one that humans would acquire from consuming mad cow. Clostridium difficile, commonly known as C. diff, in 2004 started becoming significant. This kills nearly 14,000 people each year. Ebola hemorrhagic fever and Marburg virus are both hemorrhagic fevers. So there was outbreaks of those in West Africa, still pretty significant presence there in West Africa today. 1993, cryptosporidium outbreak in the Milwaukee water supply. AIDS and HIV, they first came to public attention in 1981. What you may see here is kind of a pattern that even if we did have something to treat every infectious disease, there are new infectious diseases. There will always be new infectious diseases in the future that are going to emerge. So it's going to be a constant game of trying to keep up with them, a battle for survival between the infectious diseases and the hosts they infect.